So we start with the first one, domain bacteria, which we are very, very familiar with because in our ES syllabus, we talk about prokaryotes. So in ES syllabus, what we learn the prokaryotes, basically we refer to the bacteria. If we refer to archaeans, then there is slight difference. Okay, so bacteria, I do not need to go into very detail because we do know that it belongs to prokaryotic cells, means that they lack of membrane-bound organelles. In terms of biochemical process that normally occur in the chloroplast and mitochondria will take place in the cytoplasm of the prokaryote because they lack of the double membrane-bound organelles such as nucleus, mitochondria, chloroplast, and even in endoplasmic reticulum, they don't have. Okay, this is the features of all prokaryotic cells. In terms of the DNA, they have a circular DNA. So the circular DNA and the linear DNA is basically a very, very huge difference in terms of the replications and also the modes of the gene expressions here. Okay, so scattered within the bacterial cytoplasm, you have a many, many small loop of DNA known as the plasmid. Now you should be familiar with this plasmid, and this plasmid particularly is very important when we talk about genetic engineering. Okay, they have cell wall. Okay, so the cell wall, if they have the cell membrane as well as the cell wall, then cell wall highlights you must know it must be a peptidoglycan cell wall for bacteria. Are you clear? Okay. So again, it's a quite quick one. So you do classify the bacterial cells, feature of bacterial cells into two parts. One is the structure always present; it must be there. So peptidoglycan cell wall must be there. Plasma membrane must be there for all bacterial cells. Okay, cytoplasm, yes. Circular DNA, yes. And 70S ribosome in the cytoplasm, yes. But it depends on what kinds of the bacteria. Sometimes they may have extra structure. For example, flagellum, they may have the capsule, additional protective structure. They may have infolding of the plasma membrane. So you realize that when you have the infolding of the plasma membrane like this, it forms a sac. So this sac, can you see that? This sac basically contains the enzyme. If this bacteria can carry out the nitrogen fixation, then inside you have the enzyme for nitrogen fixation. If let's say they can carry out the process of the respiration, they don't have mitochondria, but they do have this infolding of the membrane called mesosome, and they can carry out the respiration inside, okay, or infolding to carry out photosynthesis. Okay, and plasmid is not a must for the bacteria, but generally most of the bacteria, they have the plasmid. Then they do have the pili. Pili is for conjugation process take place. Okay, for the we learn about this antibiotic resistant, right? So how two different species of bacteria they can exchange. So they exchange through this pili, something like a channel, right, to help them to establish that it is connections, then allow the transferring or sharing of this plasmid. Okay, so whenever I use this symbol, means the extra information for you, a quick one. So they are having wide diversity in terms of their morphology. They may be a spherical called cocci. They may be a rod shape called bacilli. Okay, so from this you should know already. Yesterday we learned about this Bacillus thuringiensis, right? So what is the shape? Rod shape bacillus. Can you see that? So we name them based on certain characteristics. Even though they haven't seen the bacillus before, now you do have the idea how it looks like, right? Okay, some means are vibrio. Okay, some of them will be spiral. So then will be tetrahedral or cuboidal shapes. Okay, so bacteria generally they have a single cells. They are unicellular. Okay, but they can associate to in a characteristic pattern. For example, they may in pair. Can you see that? May in pair. Okay, they may form a chain. Can you see that? In pair, sorry, in chain. Can you see that? Or they may form in a bunch. That's okay. So this is how they look like. But even though you see, is it cannot consider they are multicellular? The answer is no. Okay, they are not multicellular. Okay, because each single of cell of them, they are they're independent of other cells. So it means they have the full what we call the functionality, even though they don't have what we call they don't have other cells. Each of them they can function fully. Okay, it's not like human. Okay, multicellular organism, we have kidney, we have differentiations here, but no. Okay, so all of them, they are the same. So no differentiations in terms of into the organs, okay, tissue, and so on. So in terms of the shape, it's roughly about 0.5 to 5 micrometer in length, but some you may have a larger, but generally 0.5 to 5 micrometer. So how they reproduce? They reproduce asexually through binary fissions. Write down no mitosis, because they don't have nucleus, no mitosis. So genetic exchange may take place through the horizontal gene transfer during the bacterial conjugation process. Okay, so in our syllabus, this is the part highlight, guys. If a question asks you, okay, list or outline the characteristic feature of the bacteria, these are the items that we add in. Okay, cells with no nucleus, genetic material, circular DNA. So they don't have the histone protein associated with it. So sometimes we also call it as a naked DNA. Now, a lot of students confuse, well, naked DNA, is it means that the DNA that without or with no encapsulation of the nucleus? No, okay. Naked DNA, basically they have the DNA, they are not associated with the histone proteins. Okay, so they have circular molecule of DNA known as the plasmids. Most of the time they have, okay, no membrane-bound organelles such as mitochondria, endoplasmic reticulum, Golgi apparatus, chloroplast are present. Ribosome with 70S are smaller compared to eukaryotic cells. Ribosome, cell wall always present. It must be the peptidoglycan, not cellulose. Cell divided by binary fission, not mitosis. Can you see that? Usually exist as single cells or small group of cells, but they are still unicellular. Even though a group of cells, they are still unicellular. Okay. So these are the parts that you need to mention in the exam, particularly essay questions. Okay. List the characteristic features of a bacteria. Okay. So if you want to pinpoint out, I use different color here. What are the features specific to bacteria? Only bacteria will have these features. Here, only one. Highlight number 6, peptidoglycan cell wall. If you say no nucleus, it's not specific. Because why? Archaean later, we're going to learn, they have no nucleus as well. You talk about 70S ribosome, not specific enough. So if the question asks specific characteristic features of bacteria, only one, peptidoglycan cell wall, where you won't be able to see these features in any other living organism or other domains. Only the bacteria will have this. Are you clear? Okay, if you say no membrane-bound organelles, actually, if you look at the archaeans, they also don't have membrane-bound organelles. Okay. Another one you want to put it in is circular DNA. Okay, circular, but you have to put in not circular DNA only, does not have histone protein. This is a second one that you can write, but you must put in does not have histone protein associated with it. Circular DNA is not enough. Okay, so we have 2, but the most features, okay, because DNA, you need to check the DNA, more difficult, but you look at the cell wall, if peptidoglycan cell wall, then definitely must be bacteria. So these 2 highlights. Okay, number 2 and number 6, this is a specific, okay, specific characteristics of bacteria. Okay, so I'm very quick one, go through what are the form of bacteria that we do have, but again, it's not important, just let you know, right? We have about 30, okay, 30 phylum in the domain of bacteria. So what we do have, we have the proteobacteria, which is a gram-negative bacteria. Gram-negative bacteria was stained, okay, a bit pinkish, okay, red color. Then we do have the gram-positive bacteria, Clostridium, okay, the botulinums, the one that Botox, can you see that? Clostridium botulinums is the one that we use it for Botox. E. coli is the one that we use it for the genetic engineering. Can I see that? Okay. Then we do have cyanobacteria. Cyanobacteria is the one that can carry out the photosynthesis. So they look green color under the light microscope. Okay, then we do have the Chlamydia. It's a group of the obligated intracellular bacteria, ovoid in the shape. Can I see that? Ovoid in the shape. Then we have these spirochetes. Basically, you can see that. Okay, spiral in shape. But do we need to know this phylum? Answer is no, just for your information only. Okay, but a very, very interesting one is this cyanobacteria. We have to thanks to this cyanobacteria, okay, because before the evolutions, before the appearance of this cyanobacteria, a bit history here, okay, the whole world actually none of the organism can carry out photosynthesis. If you look at how the history of the Earth, when the Earth formed, okay, in 4.2 billion years ago, then we realize that the atmosphere actually full with the CO2, full with the CO2. So that's why C3 plants, I mean, if you look at think back about the C3 plants have no issues at all during that period of time because CO2 concentration is very, very high. And all living organisms at that time are adapted to this CO2 concentration. But if you look at the diversity of the bacteria at particular that time before this cyanobacteria starts, you will realize that the diversity is not huge, and all of them remain as the unicellular organism, no multicellular organism at all. But because due to these mutations and also this random mutation, then this bacteria evolve into having the enzyme to carry out photosynthesis, to trap the sunlight, to use the CO2 in the atmosphere and start to generate the O2 as a byproduct. Cyanobacteria is the first of them. And this cyanobacteria, what's so great about cyanobacteria? Cyanobacteria now, because CO2 is so huge of concentration in the atmosphere, so they do not need to compete with others bacteria to survive. Because why? They fully utilize the sunlight, fully utilize the CO2, so they overgrow and produce a massive concentration of O2 and nearly kill all the organism there, because O2, that means we have these redox reactions, oxidation process take place, kill many of the organism. But slowly after this, you can see that life start to emerge, more diverse. And from the unicellular, we get now multicellular, because by using the atmospheric CO2 and light energy, that is input of the energy, so it means that we can grow bigger, stronger. So that's why the diversity of the organisms start when cyanobacteria appear. So this is based on the fossil records. Now, one of the very what we call this spectacular tourist spots in Australia is this, what we call the Stromatolite. So this is cyanobacteria. Can I see the Stromatolite? So this is a fossil record to show, right? To show what? To show the evidence when these cyanobacteria start to appear in on Earth. Okay. So cyanobacteria, they are photosynthetic prokaryotes, okay, despite the fact that they are often referred to as a blue-green algae, but they are not algae, okay? So what they do, they use the water, CO2, and sunlight to create their organic carbon compound and release oxygen as the byproducts, okay? So they are first known organism to carry out photosynthesis and produce free oxygen. After about 1 billion years, the effect of this photosynthesis begins to make a huge change in the atmosphere. So the process known as the great oxygenation events, okay? So it means that the change managed to convert no oxygen in this atmosphere to nowadays 20%, okay? And because of this, the accumulations rapid during the great oxidation event as a result of cyanobacterial activity, leading to a great change in the Earth system, subsequently allowed the evolutionary emergence of the complex life. As I say that, we always start with the unicellular, and now we do have the multicellular organism, okay? And in fact, currently, if you look at these, people always say the Amazons, right, forest is a green lungs, right, of the Earth, because it contributes to huge concentration of the O2 on the Earth. But in fact, even though we say the Amazon's forest, the tropical forest, give us so much oxygen, but in fact, bacteria is still the one that give us, the cyanobacteria is still the one that give us, okay, and the plankton in the oceans give us about 70% of the oxygen on Earth. Okay. So we come to the next one. So stop the recording.